Okay, let's start on chapter 17, blood. All right, you need to understand the anatomy and physiology of blood. That's going to help you advise patients on activities that prevent blood clots during the time that they're in hospitals. Makes perfect sense. Um, Here's the video. We'll see if this works, like if it records properly. If it doesn't, that's okay. You have access to this PowerPoint yourself. You can look this up. Okay. So you understand that blood is a transport vehicle. That's what they just went through. What we're talking about is that it's carrying oxygen, carrying nutrients to the cells, carrying their waste, their CO2 away. Um, that we will then breathe the CO2 out in the lungs or use kidneys to remove waste from the body. So functions of blood: transport, regulation, protection. We just talked about transport. Regulation: blood is involved in regulating temperature, regulating pH. Protection: clotting and the whole delivery of white blood cells. So clotting will, the coagulation, we are going to prevent blood loss. We are going to close wounds in the body. And the delivery of white blood cells, obviously, those are the soldiers of the immune system. Anywhere you deliver them, you're protecting the body from outside invaders. All right, so transport. Like I said, O2 and nutrients are going to the body cells. Metabolic wastes that you remove are going to be taken to the lungs. We're talking about CO2 to breathe out. To the kidneys, we're talking about things like urea for you to eliminate. And it also is used for, this is something I didn't mention before, transporting hormones through the body from where they're produced to the target organ. So it's a way of sending messages and signaling throughout the body, helping to coordinate the actions of different organs throughout the body. All right, regulation. I already said we use it for temperature, we use it for pH. Fluid volume is something I hadn't mentioned, maintaining adequate fluid volume. So how does that matter? Well, if you don't have enough fluid volume, you're going to have trouble delivering the blood to the brain, which means you're going to have trouble delivering oxygen to the brain, and the brain needs a lot of oxygen. All right, protection. I already said preventing blood loss. I know you're like, how could blood protect against blood loss? But the reason is because the blood is full of proteins, full of thrombocytes, which we also call platelets, that will help make clots and prevent further blood loss. So yeah, the blood is preventing blood loss, as weird as that sounds, because the blood contains things that will make clots and keep most of the blood in. And preventing infection, we're carrying antibodies, we're carrying white blood cells, we're carrying complement proteins. Complement proteins are amazing. They will go up to a bacteria and literally rip a hole into the membrane so the bacteria dies. Bacteria spills all its guts out. Really neat little part of the immune system. All right, composition of blood. Blood is considered to be a tissue. Most of it is plasma, the fluid that's not alive. The rest of it, the living things, are the formed elements. And they say all the living blood cells are formed elements, but if you look below where it says platelets, I want to emphasize that platelets, thrombocytes, are not cells, they are pieces of cells. I know it's a minor deal, but we'll get to it. So formed elements are red blood cells or erythrocytes. Urethra means red. Leukocytes or white blood cells. Leuko means white. And platelets, also called thrombocytes. Now, we're going to have to learn about the different types of white blood cells in a little bit. I'm going to go ahead and give you a mnemonic to use for that. The different types of white blood cells, we remember that by saying never let monkeys eat bananas. Never let monkeys eat bananas. So N for neutrophils, L for lymphocytes, M for monocytes, which will grow up to be macrophages. So M for monocytes, E for eosinophils, B for basophils. That's not just the types of white blood cells. That's the types of white blood cells that in a healthy body are in proportion, in order, from most common to least common. One of the tests, the clinical tests that you will do in a lab in a hospital, is to see if those ratios are off. If you have a lot more basophils or eosinophils, then you know what type of problem is going on in the body. So yeah, different types of white blood cells are specialized for different types of things, fighting different things. And if you see a rise in the percentage out of the normal range, you can probably guess based on that what kind of problem the body is having. All right, when we take the blood and we spin it down in a centrifuge, they have these little tiny, they fill up a microcapillary tube with blood, and then they spin it in a microcentrifuge or a hematocrit centrifuge. What you're doing is separating it out, so you'll have the plasma towards the middle of the centrifuge. Usually that's the top of the tube. You will have at the bottom of the tube all of the red blood cells, that's most of it. And in the middle, this tiny little layer is the buffy coat. That is all the thrombocytes, that is all of the white blood cells. So anyway, the amount of red blood cells, the erythrocytes that are to the outside, to the bottom of the tube, we'd expect that to be around 45%. In males, 47 is normal, plus or minus 5. Females, 42 plus or minus 5. The buffy coat should be less than 1%. So only less than 1% of your blood is white blood cells and thrombocytes, platelets, to help with clotting. The plasma on the top will be around 55%. So, all right. So there's a pretty picture of it all spun down. You understand. Got the numbers that they gave you. You should know those numbers. Let's move on. So blood is sticky, is opaque. That means you can't see through it. It's not clear like water. And tastes metallic. I know that sounds weird, but yeah, they did used to diagnose things in the body in the olden days through taste. Why would it taste metallic? because of the iron. Remember, hemoglobin has hemes in it. So all those red blood cells have hemoglobin, and that has heme groups in it, and heme is iron. Heme has iron. It says the color of blood varies with oxygen content. Totally true. High oxygen red. High oxygen is a really bright red. We're calling it scarlet. I call it like a bright neon cherry kind of red. Low oxygen blood is more of a brick red, a dark red. So, and we have names for that. We call the hemoglobin that is causing the bright red color, it's full of oxygen, oxyhemoglobin, and we call, not surprisingly, the hemoglobin in the dark brick red, the one that's low in oxygen, call that deoxyhemoglobin. Why is there a difference? Because the heme actually changes shape. It'll grab onto the oxygen and bend around it to hold it tight. And when it does that, the change in shape, that little tiny change, changes which wavelengths of red get reflected back. Lead has a pH of 7.35 to 7.45. That's a little on the alkaline side, a little above neutral, but it's pretty close. It makes up 8% of the body weight. You've heard that. It was in that little video on the second page of this chapter. The average volume in males is 5 to 7 liters of blood for the human body, and for females, 4 to 5. The difference is only based on the size. There are a lot of women who are smaller than men, and so women have slightly smaller bodies, slightly smaller amounts of blood in the body. Okay, blood plasma. Remember, that was the 55% right there. Okay, blood plasma, it's sort of yellowish. Okay, straw colored, sticky. It's mostly water, 90% water. Has a In that water, you have more than 100 different things dissolved. Remember, water is the solvent. The things that are dissolved in it are the solutes or solutes. Yeah, anyway. I was trying to draw that out to emphasize it, and it comes out about the same. But solute and solvent. Solvent is what you dissolve things in. We dissolve things in the water. Solute is what you're dissolving. If you remember from AP1, I compared this stuff to I compared a solution to a refreshing Kool-Aid drink. That's a solution. We have the solvent, that's the water, and then we have the solutes, that's the sugar and the Kool-Aid powder that we dissolve. Blood has more than 100 different things that we dissolve in it. It could be nutrients, gases, hormones, waste, proteins, inorganic ions. There's some overlap and redundancy there when they say waste. Some of those are gases, like the CO2. Anyway, these solutes are some of them are plasma proteins, and they are the most common thing. Some of these, like albumin, really big. When the blood gets to the tissues and then going through the capillaries, offloading oxygen and picking up carbon dioxide, those plasma proteins stay in the blood vessel. They don't move around. So, yeah, they're staying in the blood. They're not leaving the capillaries and going around the cells. These proteins are mostly made in the liver, and albumin is 60% of the plasma proteins. Albumin is huge. Now, when we say albumin, you might say, but I've heard of egg albumin. Yeah, you have. Yeah, totally. Albumin is just a family of round globular proteins. And there's egg albumin, there's also blood albumin. And different animals are going to have different types of blood albumin. They're going to have slightly different shapes or slightly different sequences of amino acids. Albumin can work as a carrier for other smaller molecules, including oxygen or carbon dioxide. It's not just a red blood cell thing carrying it around. And it can act as a blood buffer. Why? Well, because one of the things that it can carry around is carbon dioxides or hydrogens. And of course, those are involved in pHs. And it contributes to plasma osmotic pressure. That's a fancy way of saying that water wants to dilute stuff. And if the water at the beginning of a capillary flows out of the blood in the capillaries, the albumin stays behind and the blood gets more concentrated, more concentrated, more concentrated. Eventually, that water wants to flow back into the capillary to dilute all of the albumin that's becoming more and more concentrated. The wanting to come back into the capillary is what we call osmotic pressure. So, anyway. All right, so here is a table that talks about what is in blood. This is table 17.1. Definitely know this table. All right, 17.1 part 2. Again, we're talking about what's in the blood plasma. So now we've gone through plasma. Let's talk formed elements. Remember, the formed elements are the living things. That's the red blood cells, erythrocytes, the white blood cells, the leukocytes. That's all of them. Never let monkeys eat bananas. So neutrophils, lymphocytes, monocytes, eosinophils, basophils. It's also the thrombocytes or platelets, which help with clotting. It says only white blood cells are complete cells. Totally true. It says red blood cells have no nuclei or other organelles. You need to know that's an adult red blood cell. When the young red blood cell is forming, as it grows up, it sheds the nucleus. It gets rid of the nucleus. It gets rid of almost all the contents except hemoglobin. Why? Well, because say you had a bunch of mitochondria inside, they're going to need oxygen. They're going to use oxygen. Red blood cells' job is to carry oxygen. So if you get rid of everything else, you're not going to waste the oxygen on keeping the cell equipped with oxygen. Instead, that red blood cell is just going to carry that oxygen around for use elsewhere in the body. So no nuclei or other organelles because we shed those as the red blood cell grows up. Platelets, thrombocytes are cell fragments. Absolutely true. They are not a complete cell. You have a cell that they pinch off of, and those are the important blood clotting things. But they're not a true cell. They're pieces of a cell. Hold on a second.